Welcome back friends. Welcome back to another video session from this channel. So in this video session we will be studying regarding the various raw materials which are required for organizing the transcription mechanism or the transcription process. Is it okay? So what are the various raw materials which govern the transcription mechanism or the transcription process? So the various raw materials which are required for organizing the transcription mechanism or the transcription process include promoter region. Is it okay? Promoter region, template stand, RNTPs and RNA polymerase. So all these are the various raw materials which govern the transcription mechanism or the transcription process. Is it okay? So at first we are going to study regarding the promoter region. So what is promoter region or how we can define the term known as promoter? So likewise in case of the replication mechanism, in what way? For initiating the replication mechanism or the replication process, the specific mechanism or the specific process, it takes the advantage of a specialized region which is a part of the double-stranded genomic sequence, which is termed as origin of replication, which is simply termed as ORIC. Is it okay? In the same way, for initiating the transcription mechanism or the transcription process, the specific mechanism or the specific process, it takes the advantage of a specialized concerned nucleotide sequence within the double-stranded genomic sequence. So the specialized concerned sequence which is responsible for initiating the transcription mechanism or the transcription process, it is nothing but what? Promoter region. Is it okay? So by taking the advantage of promoter region, here the transcription mechanism or the transcription process, it is successfully initiated. So here if we consider the specific promoter region, the specific promoter region basically it consists of two different consonant sequences. Is it clear? So for your better understanding, let me present it on the board. So this is the 5' end. Is it okay? So here there is an availability of consonant sequence termed as TTGACA. Is it clear? And here there is an availability of one more other consonant sequence TTAA TAT. So let me erase the things here. So this is the 3' end. Is it clear? So here the complementarity sequence of this particular strand, it relates in case of the other strand. So it is the 3' end. So here the complementarity of T is A, again it is A, C, T, G and T. And again the complementarity of T is, it is A, T, T, A, T and A. Is it okay? And it is the 5' end. So this is termed as consonant sequence. Is it okay? So what is the meaning of consonant sequence or how we can define the term known as consonant sequence? A sequence of common nucleotides which are most commonly found in the homology region of the double-stranded genomic sequence, it is termed as what? Consonant sequence. So if we look at here, this particular sequence, it is rich in thymine and adenine nucleotide. So the reason why this particular sequence, it is termed as consonant sequence. So this particular consonant sequence or it is otherwise termed as Tata box. So why it is termed as Tata box? Because this particular sequence, it is rich in thymine and adenine nucleotide. So the reason why this particular consonant sequence or it is otherwise termed as Tata box. Is it clear? So let me write it here. Tata box. So here this particular consonant sequence which is termed as Tata box. So the other term for this Tata box is Pribno box. Tata box, or it is otherwise termed as Pribno box. So now here my question is, why this particular consonant sequence it is termed as Pribno box? Because for the very first time this particular consonant sequence it was identified, it was observed by a well-known molecular biologist David Pribno. So on the behalf of his discovery, this particular consonant sequence it was termed as Pribno box. Pribno box or Tata box, anything is same. Is it clear? So let me write it here. Pribno box. So likewise this particular consonant sequence, if you look at here there is an availability of one more other consonant sequence, and this particular consonant sequence it is rich in TTGACA. Is it clear? So the particular consonant sequence it is rich in TTGACA, but unlike this particular consonant sequence like Tata box, this particular consonant sequence it do not possess or it do not contain any consonant names like Pribno box or Tata box. So simply it is termed as consonant sequence. Is it clear? So this particular consonant sequence it is rich in TTGACA. So the reason why this particular sequence it is termed as consonant sequence. So in addition to the consonant sequence, very nearer to the promoter region, if you look at here there is an availability of a specialized site, and that specialized site it is nothing but what? Transcription start site. Is it clear? So let me arrange the things here. Transcription start site. Is it okay for you? So why it is termed as transcription start site? Because when the RNA polymerase enzyme, when it successfully recruits to the promoter region, the very first RNTP which is successfully recruited during the transcription mechanism or the transcription process. So simply it is denoted as what? +1, or in other terms we can define it as transcription start site. So from the point of transcription start site, is it clear? From the point of transcription start site, this particular consonant sequence it is located around 10 nucleotides far away. So the reason why this particular consonant sequence it is termed as what? -10 consonant sequence. Is it clear? So let me write it here. What it is? -10 consonant sequence. So in the same way, from the point of transcription start site, the other consonant sequence it is located around 35 nucleotides far away. So the reason why this particular consonant sequence it is termed as -35 consonant sequence. Is it clear? -35 consonant sequence. And the distance between -10 consonant sequence and -35 consonant sequence it is around 16 to 18 base pairs. Is it clear? So here during the transcription mechanism, at the very beginning of the transcription mechanism, in order to initiate the transcription mechanism or the transcription process, the RNA polymerase enzyme which is processive in nature, it has to recruit to the promoter region. But the specific RNA polymerase enzyme itself, it do not have any higher affinity to recognize the promoter region. In order to recognize the specific promoter region, the RNA polymerase enzyme, it takes the advantage of a specific factor which is nothing but what? Sigma factor. By taking the advantage of a specific factor termed as sigma factor, RNA polymerase enzyme, it acquires higher affinity to recognize the promoter region. Is it clear? So let me write it here. What it is? Sigma factor. And the specific sigma factor which is most commonly used in order to initiate the transcription mechanism or the transcription process in case of prokaryotes, it is sigma 70. sigma 70. So here you might assume that in all the transcription mechanism or in all the transcription process, the specific sigma factor which is used, it remains as sigma 70. But this is not the case. The specific sigma factor, it remains vary from organism to organism and from species to species. In all the transcription mechanism or in all the transcription process, the specific sigma factor value, it won't remain as sigma 70. It remains vary from organism to organism and from species to species. And now here my question is, why the number sigma 70 is given to this particular factor? Because the molecular weight of the sigma is around 70. So the reason why the particular sigma factor, it is termed as sigma 70. So right now we are not going to study, we are not going to discuss regarding the sigma factor. In the next upcoming video session, surely I will explain you what is sigma factor and how the specific sigma factor, it plays a prominent role in order to recognize the promoter region, and what are the various subunits of sigma factor and what are the various types of sigma factor in detail. Is it clear? So in the next upcoming video session, surely I will explain you all these things in detail. So right now we are not much more concerned regarding the sigma factor. So this is regarding the specific promoter region. So here I need to mention you one key point here. The specific promoter region, it is termed as cis-acting element. Is it clear? The specific promoter region, it is termed as cis-acting element. So let me write it here. What it is? Cis-acting element. Cis-acting element, right? So here, why it is termed as cis-acting element? Why the specific promoter region? Why it is termed as cis-acting element? That means when the specific RNA polymerase enzyme, when it successfully recognizes the promoter region. Is it clear? When the RNA polymerase enzyme, when it successfully recognizes the promoter region by taking the advantage of sigma factor, the specific RNA polymerase enzyme, it won't encode the information which is present in the promoter region. Instead of encoding the information which is present in the promoter region, the specific RNA polymerase enzyme, it encodes the information which is present in the rest of the genomic sequence. That means the specific promoter region, it is termed as non-coding DNA. Because the RNA polymerase enzyme, it won't encode the information which is present in case of the promoter region. Instead of encoding the information which is present in case of the promoter region, the RNA polymerase enzyme, it encodes the information which is present in case of the rest of the genomic sequence. So the reason why the specific promoter region, it is termed as cis-acting element. Is it clear? So this is regarding the specific promoter region which is responsible for initiating the transcription mechanism or the transcription process. And the next specific raw material which plays a prominent role for organizing the transcription mechanism or the transcription process, it is nothing but what? Template DNA. Is it clear? So by taking the advantage of template DNA, here the transcription mechanism or the transcription process, it is successfully organized. But now here my question is, out of the two strands of DNA, which particular DNA strand, it acts as template for organizing the transcription mechanism? In case of the replication, is it clear? In case of replication, out of the two strands of DNA, so these are the two strands, right? So out of the two strands of DNA, both the strands of DNA, they act as template for organizing the replication mechanism or the replication process. But whereas in case of transcription mechanism or in case of the transcription process, out of the two strands of DNA, only one particular strand, it acts as template for organizing the transcription mechanism or the transcription process. But now here my question is, out of the two strands of DNA, which particular strand, it acts as template for organizing the transcription mechanism? Whether the DNA strand which is in 5' to 3' orientation or the specific DNA strand which is in 3' to 5' orientation. So the answer to this question is very simple. The specific DNA strand which is in 3' to 5' orientation, it acts as template for organizing the transcription mechanism. Is it clear? The specific DNA strand which is in 3' to 5' orientation, it acts as template for organizing the transcription mechanism. So here you may get a small clarification why the specific DNA strand which is in 3' to 5' orientation, it acts as template for organizing the transcription mechanism. Because Likewise, the replication mechanism, the specific transcription mechanism or the transcription process, it proceeds in 5' to 3' direction. That means in order to synthesize the nascent mRNA during the transcription mechanism or the transcription process, the specific template DNA, it should remain in 3' to 5' orientation, but not in 5' to 3' orientation. Is it okay? If the specific template DNA, if it remains in 3' to 5' orientation, under such condition, the nascent mRNA, it is successfully synthesized in 5' to 3' orientation. If the template DNA, if it is 5' to 3', under such condition, the nascent mRNA, it remains in 3' to 5' orientation. So, which is not at all acceptable, right? So, the reason why the specific template DNA, it should remain in 3' to 5' orientation, and the nascent mRNA, it is successfully synthesized in 5' to 3' direction. Is it clear? So now here my question is, the specific DNA strand which acts as a template, it is termed as template DNA, or in other terms we can define it as antisense strand. The specific DNA strand which acts as template for organizing the transcription mechanism or the transcription process, it is termed as template DNA, or in other terms we can define it as antisense strand. And the specific DNA strand which do not act as template for organizing the transcription mechanism or the transcription process, it is termed as sense strand or coding strand. Is it clear? So let me write it here. What it is? Template strand. Template strand or antisense strand. Is it clear? And the specific strand which do not act as template for organizing the transcription mechanism or the transcription process, it is termed as sense strand or coding strand. So why it is termed as sense strand or coding strand? Because the nascent mRNA which is going to synthesize in the transcription mechanism or in the transcription process, it is quite homologous to the specific DNA strand which is not acting as a template. So the reason why the specific DNA strand which do not act as a template for organizing the transcription mechanism or the transcription process, it is termed as sense strand or coding strand. Is it clear? So let me write it here. What it is? Sense strand. So let me use some other color here. Sense strand, or in other terms we can define it as coding strand. Is it clear? Now, so many of the students, they confuse here. Many of the students, they assume that here the RNA polymerase enzyme, it proceeds in 5' to 3' direction and it synthesizes the mRNA in 5' to 3' orientation. So this is an incorrect strategy. The specific RNA polymerase enzyme, after the successful recognition of the promoter region by taking the advantage of sigma factor by the RNA polymerase enzyme, the specific RNA polymerase enzyme, it proceeds in 3' to 5' orientation. Is it clear? The specific RNA polymerase enzyme, it proceeds in 3' to 5' direction and it incorporates the RNTPs in 5' to 3' orientation. Is it clear? So many of the students, they assume that here the RNA polymerase enzyme, it proceeds in 5' to 3' orientation and it incorporates the RNTPs in 5' to 3' orientation. So it is an incorrect strategy. RNA polymerase enzyme, it proceeds in 3' to 5' orientation and it incorporates the RNTPs in 5' to 3' orientation. Is it clear? So let me present it here so that easily you can understand. 5' to 3' and 5' to 3'. So here it is the directionality and it is the incorporation of RNTPs, right? So here the specific RNA polymerase enzyme, it won't proceed in 5' to 3' direction. It proceeds in 3' to 5' orientation and it incorporates the RNTPs in 5' to 3' orientation. So the reason why it is an incorrect strategy, right? And the correct strategy is RNA polymerase enzyme, it proceeds in 3' to 5' orientation and it incorporates the RNTPs in 5' to 3' orientation. So it is the correct strategy. So this is regarding the template strand. And the other important raw material which plays a prominent role for organizing the transcription mechanism or the transcription process, it is nothing but what? RNTP. Is it clear? So let me present it here. So let me arrange the things first. So what are RNTPs? RNTPs, right? Is it clear? So the specific nucleotides which play a prominent role for organizing the transcription mechanism or the transcription process, they are nothing but what? RNTPs. In what way? For organizing the replication mechanism or the replication process, DNTPs play a prominent role in the same way. Is it clear? In the same way, for organizing the transcription mechanism or the transcription process, RNTPs play a prominent role. But now here my question is, what is the basic difference in between DNTP and RNTP? So for your better understanding regarding the basic difference in between DNTP and RNTP, so let me present you the structure of RNTP so that easily you can understand what is the basic difference in between DNTP and RNTP. Is it clear? So let me present it here. So this is the purine ring structure, right? Purine ring structure. Is it okay? And here it is nothing but the ribose sugar. Ribose sugar. So this is second hydroxyl group and it is the third hydroxyl group. And here it is nothing but the CH2 group, right? And here there is an availability of phosphate moiety. So this is nothing but what? The structure of RNTP, right? So what is the basic difference in between RNTP and DNTP? So in case of DNTP, there is an availability of hydroxyl group only at the third position. Is it clear? So in case of DNTP, there is an availability of hydroxyl group only at the third position. But at the second position, instead of having hydroxyl group, there is an availability of only one hydrogen atom. So the reason why it is termed as what? Deoxynucleotide triphosphate. That means lack of oxygen atom at the second position. So the reason why it is termed as what? DNTP. But whereas in case of RNTPs, there is an availability of hydroxyl group both at the second position and as well as at the third position. So the reason why it is termed as what? Ribonucleotide triphosphate. Is it clear? So here, why it is termed as ribonucleotide triphosphate? So what is the meaning of nucleotide? Nucleotide, it is nothing but what? It is an association of nitrogenous base, sugar and phosphate moiety. Is it clear? So it is nothing but what? Nitrogenous base, and it is the sugar and it is the phosphate moiety. So the reason why it is termed as nucleotide, and why it is termed as triphosphate? Because it possesses 3 phosphate moieties. So these are the 3 phosphate moieties, right? So the reason why it is termed as ribonucleotide triphosphate. So what are the various RNTPs which play a prominent role for organizing the transcription mechanism or the transcription process? So the various RNTPs which play a prominent role for organizing the transcription mechanism or the transcription process include RATP. Is it clear? RATP, RUTP, RGTP and RCTP. So let me present it here. What are those? RATP, RUTP, RGTP, and the last one is RCTP. So here I need to mention you one thing. So here in case of the DNA, there is an availability of thymine, but whereas in case of RNA, there is no availability of thymine. Instead of having thymine, there is an availability of uracil. So the reason why for the successful synthesis of RNA during the transcription mechanism by the RNA polymerase enzyme, the specific mechanism or the specific process, it takes the advantage of RUTP but not RTTP. Is it clear? In case of RNA, there is no availability of thymine. Instead of having thymine, there is an availability of uracil. So the reason why the specific mechanism or the specific process, it takes the advantage of RUTP but not RTTP. So this is regarding the specific RNTPs which play a prominent role for organizing the transcription mechanism or the transcription process. And the next specific raw material which play a prominent role for organizing the transcription mechanism or the transcription process, it is nothing but what? RNA polymerase enzyme. So which we are going to study, which we are going to learn in the next upcoming video session. So in the next upcoming video session, surely I will explain you what is RNA polymerase. So what are the various subunits of RNA polymerase in case of prokaryotes, and what are the various types of RNA polymerase which are used in case of eukaryotes. So I hope that this video will help you a lot. So this is regarding the various raw materials which are used in case of the transcription mechanism or in case of the transcription process. So I hope that this video will help you a lot. So if you like this video, just hit the like button and share it to your friends. And I remember you people to subscribe my channel for getting more and more videos what I make day to day. Thank you.